Hi, it's Mr. Rikyu, and today we're going to talk about B3.1 gas exchange. So the main objective is to analyze how gas exchange works in humans. Okay, we're also going to talk about plants and fish, but the main goal, the main objective is to examine how it works in humans and the potential problems. Okay, the causes and consequences of many diseases and problems that we face. Okay. Um, in fish, how it looks like. It looks like the gills. We're gonna see some diagrams, some images like this. Probably when you go, if you go to the supermarket to buy a fish, eventually you might see people cleaning the fish. Okay, and then you're gonna see the gills that are these layers in which gas exchange happens. All right. So before we start, something to think about is the highest to lowest rate of transpiration, of exchange of gases, exchange of molecules between these environments. Among these options, what do you think we would have highest or lowest rate of transpiration? So consider the temperature and also the levels of moisture. Okay. Okay, so the first concept that we need to cover is the surface area to volume ratio. All right. This is something that we cover in cell structure and cell biology, but is also going to matter here. Okay. In other words, the smaller you are, okay, and the smaller your volume, and especially the surface area to volume ratio, the smaller it is, the fast it is to exchange materials between the surface and the outer environment. All right. And the bigger you are, the more challenging it is. This is the unique mathematical and physical explanation why cells are small. This is why there are no cells that are large and huge. Okay, because it's very difficult to exchange the molecules from the interior to the exterior. All right. Imagine, for example, if we had a classroom and this classroom is massive, like a soccer field. It would be very hard for the students to get in and get out. Would take time for you to go to the bathroom or to drink water. The same thing here. And as you increase the volume of um, this surface, the surface area to volume ratio drops quickly, and it's hard to interact with the outer environment. All right, so it's more efficient to be and to stay with a smaller volume. All right. So first, let's check some summary points. Okay, things, let's say structures that are needed for you to have proper gas exchange. Okay, we're going to cover all of these. So large surface areas, permeable membranes, thin tissues, moist linings, and a maintenance of concentration grade. I'm going to explain each one of them. All right. So first, large surface area. Why large surface area? We need this to have a proper gas exchange. All right. If you have just a flat surface, things are the area that is in contact with the environment. It's not that large. Okay, it's just a flat 2D structure. Once you create infoldings and folds, you increase the potential area that is touching the environment. And this is what we do. This is what fish do with their gills. Okay, you have several layers, okay, enfolding tiny thin filaments in which water is going to pass, and then you can have a proper gas exchange for the fish. All right, this is the first concept. This is what we see here. So we have these layers and then full of capillaries, full of arteries and tiny veins, and then you can have a proper exchange. If it's just like a flat or a circular surface, you would have exchange only on the surface, but not in the interior. So it wouldn't be efficient. These fish, they wouldn't exist. Next one, and we have permeability. So what is permeability? Basically is the possibility of being permeable, of materials, gases, and molecules to pass through, to pass and be exchanged. All right. So, for example, a surface that is not permeable, unpermeable, is our skin. If you put your skin in the seawater, the seawater is not going to get in. Okay, salts are not going to get in your body because it's impermeable. Nothing passes here. All right. But otherwise, your mucosa or your inner layers, or for example, your stomach or your lungs, they're permeable. Once molecules arrive in this structure, they pass through the membranes and they reach the cells. Okay? Here we have an image of stomata and leaves of plants. Okay, so then we have exchange of gases. Next, we have thin tissues. So for materials to move, they need to be thin. Okay, so for example, if you are in a very cold place, you wear a jacket, and that jacket is not letting you exchange heat, exchange air with the outer environment because it's thick. Okay, it's big, it's fixed, it's thick, so it's not letting things pass. Otherwise, if you're using a t-shirt in winter in New York, what's going to happen? It's thin, it's permeable, it's just cotton. So the air and the heat is going to be exchanged. All right. So the thinner you are, the more exchange you have. Okay. This is what happens in the lungs. So here we have some alveoli, and we can see the thin layers that are surrounding these structures. So then you can have proper gas exchange. Okay. Good. Moist lining is the next one. So moist lining is basically these surfaces being surrounded by fluids that maintain their movement, okay, and don't let them collapse. Okay, if you have these surfaces that are very thin and very permeable, if they get dry, they collapse. Okay, so for example, when you are in a pool with your hair below the water, your hair is floating. Once you leave the water, they collapse. This is what happens here. And we avoid by producing moisture, by producing surfactant, and they create this moisture that prevents this. And next we have concentration gradients. How do we maintain concentration gradients? By constantly supplying the lungs or the gills with blood, okay, with deoxygenated blood, and constantly supplying these structures with gases. In the case of mammals, us, we're constantly breathing in and out and sending oxygen and sending gases to the lungs. In the case of fish, they're constantly moving water towards the gills. So then this physical exchange of gases can happen. Um, so again, we need a permeable surface, okay, for these materials to pass. And first, to have this concentration gradient, we need a dense network of blood vessels and continuous blood flow with ventilation. This is how we maintain this forever and ever in our lives. Okay. Good. So checking the mammalian or the human lungs, this is the typical structure. Okay, this is the basic structure. We have our mouth. Okay, and then we have the trachea that the gas is going to pass. Nose, trachea, and from the trachea, this is going to move to the bronchus and fall into the lungs. Okay, in the lungs, you're going to have the exchange. Here in the lungs, you're going to branch over and over and over until you reach the alveoli, that are these tiny structures that are very thin, very delicate, and the gas exchange is happening here between the capillaries and the alveoli. Okay. Um, and the surfactant that is present in these structures, okay, they're produced by the pneumocytes number 2, type 2 pneumocytes. They secrete these fluids to maintain the cells alive and moving, so then they don't collapse, they don't get dry, and they don't collapse. The pneumocytes type 1, they're basically involved in the gas exchange. They're very thin and flat, and they're involved in the gas exchange. This is important for the IB exams. All right, so the principle, the idea is the same. It's always branch, branch, branch. Reduce the size to increase the surface area. Okay, so reduce the surface area and increase the surface area to volume ratio, right? And of course, for us to get this oxygen, we need this extensive capillaries running through the alveoli to exchange, deliver the CO2 to the alveoli and get the O2 from the environment. So then you can send this to the whole body. Observing this in micrographs, in IB papers, or in microscopes is not that simple, okay, because these tissues are thin. So we end up observing these tiny sacs, these layers, okay, and these are the alveoli, and the oxygen, and the gases, and the capillaries, they're passing through here. They're basically passing through, but it's hard to observe. And we need tons of these. We have thousands of these alveoli, right? You don't have just one. We have many, many of them, and they're very small. And is where we exchange our gases, right? Okay, so problems, diseases associated with lungs. Okay, the first one is emphysema. Emphysema is when people smoke like vape, pipe for many years, and these toxins, these smokes, these fogs, these substances, they damage the wall of the alveoli. By damaging, it will change the surface, or they create a more thicker layer. In other words, you end up changing this. You change the structure, and you reduce the efficiency. You reduce the efficiency of gas exchange, and this is highly problematic because basically you need to make more force to get the same amount of ventilation in your lungs. This is why typically you won't observe people, smokers, working out a lot or running marathons or biking, cycling, or swimming a lot because it's hard for them. They need to make more force to get the same amount of air that you, non-smoker, that you have in your lungs. Next one is lung cancer. Okay, lung cancer is basically a cancer. Okay, so we can have cancer in any kind of tissue in our body, but in this case, in the lung, and especially due to pipes, cigars, or narguilés, okay, or cigarettes that will end up creating mutations in a specific group of cells that can develop into a tumor and then harm the lungs or eventually harming the whole body. Um, Okay, one activity interesting for you that we can do is to draw a label diagram of the alveoli and surrounding blood vessels. This is possibly covered. Okay, it is very possible to be covered in paper 2 examinations, standard level or higher level. All right, so be able to label these structures or draw these structures, create these diagrams. It's interesting. It's going to help you on paper 2s. Okay, moving forward, we have ventilation in the lungs. We're almost finishing the center level component of B3.1. Ventilation in the lungs is mainly created by the diaphragm, okay, and the muscles, the intercostal muscles and the ribs, okay, as they increase and decrease the space, and as they increase and decrease the pressure, you're going to ventilate, breathe in and out, okay? So when we contract the, when we relax the diaphragm, okay, so you are going to reduce the space in the lungs and increase the pressure. So then you are exhaling. When the opposite happens, when the lungs are expanding due to the muscles, when the lungs are increasing its volume, you reduce the pressure, so then air gets in, and then you are inhaling the air. Okay. We can measure lung volumes by a series of tools. Okay, the most common ones are the spirometers. Okay, these are techniques, these are devices that can be definitely covered in IB papers. Okay, so paper 1, paper 2, paper 1B, they can be covered, and some questions might be asked about it. The spirometer basically is used to measure the lung volume. Okay, the volume in liters of air that can be uptaken by an individual. Okay, and we end up measuring some statistics, for example, total lung capacity or vital lung capacity or other sorts of lung capacity. All right, and we have these graphs that are definitely not hard to to observe and to interpret. Okay, so here we have some samples. We have tidal volume, that is a natural flow, normal breath. We have the inspiratory reserve volume, so the maximum that you can breathe. We have expiratory reserve volume, and we have also residual volume. The residual volume is the amount of air that typically always stays in your lungs. We never completely empty our lungs. All right. Going to plants. Okay. Feel free to pause the video to come back to the video or to stop in a specific place of the video that you want to study more. Here I'm going to continue until the end, go over all this in a level higher level, but feel free to stop or to repeat in a section that you prefer. So continuing, now we're going to talk about plants. Okay, so in plants, we also have tubes. We have vascular bundles or veins, right? Especially the xylem and the phloem. The xylem transports typically water and minerals, and the phloem water with sugars, carbohydrates, okay, fructose, sucrose, okay, and also amino acids. The xylem tends to stay, for example, here in the leaf, stays the center of the leaf, and it's more structured and larger and more lignified than the phloem. Okay? How a leaf is adapted for gas exchange. This is important. Uh, First, they're thin, they're flat, so this increases the surface area to volume ratio for gas exchange. And they also have openings. Okay, they have the openings that are the stomata. They need these openings to exchange the gases. So when they open, they release a vapor of water and they end up uptaking CO2. They need to do this. Otherwise, they don't get the CO2 that they desperately need for photosynthesis. So when they open, they lose a little bit of water, but they get the CO2. So it's a trade that they constantly need to efficiently balance in their lives. Okay, so they're thin, it helps, but they also have the stomata that are these openings, and the guard cells that regulate this, and this is how they uptake the CO2. Okay, the upper epidermis and lower epidermis, we have protections against dehydration. Okay, so imagine a plant, tree, in a farm or in a savanna, they can simply lose water. They're not permeable. They have protections, but they have the stomata to open these pores and to exchange properly when needed. Okay, if there is a constant supply of water, if there is no shortage of water, then basically they won't close. They won't simply close because they want more CO2. But if there is some shortage of water, there are some strategies that different groups of plants end up applying to manage this and to navigate this challenge. Okay, great. So the stomata again is a pore that is regulated by the guard cells. Okay, this is a picture. This is a real picture. It's beautiful. Okay, so we have this like this mouth structure, and then depending on how the plant regulates the osmolarity of the guard cells, this is going to open or close. Okay, this is especially important exchange of water vapor and CO2. Without this, plants can't get CO2, and without CO2, they don't do photosynthesis and they don't live. All right. Just to close the part of plants, we have the sponge mesophyll, again, phloem and xylem in the center. The xylem is more lignified and it's more visible. And we have the epidermis. Okay, we call this the vascular bundle in plants. Great, interesting part. Some experiments that I use to measure transpiration. Okay, this is especially possible to be present, okay, in paper 1Bs, but could also be in paper 2 or paper 1. But paper 1B are interesting for this. This is a potometer, okay? Potometer is a simple device that can measure transpiration rates in plants. So basically, you take a stem, you place in a tube, like glass, for example, with a cork, and then you seal here, and you're going to have water with a volume scale and an air bubble in a reservoir. What happens? These plants will start to evapotranspirate through the xylem, okay? And as they get water, this bubble or the volume of water is going to decrease. And some factors might increase or decrease this transpiration. This is what we study in a potometer and in these labs. So moisture, temperature, light, and wind intensity is definitely going to influence, increase or decrease the transpiration rate of these plants. Okay, so for example, if you have more wind in this shoot in this stem, you're going to increase the rate of transpiration, the rate of exchange. The plant's going to have more transpiration, release more water, and get more CO2. If you have less wind, less transpiration. Light intensity, same thing. More light, more transpiration, because plants are going to make more photosynthesis. Temperature, same thing. Okay, if you have more heat, higher temperatures, more dynamic in the molecules, more thermal energy. And the plants are also going to make more metabolism. So more photosynthesis, they end up transpirating more water to the environment. But if we have humidity, if we have moisture, it's the opposite. If you have a bunch of moisture, if you put a plant like in a sauna, what happens? You have so much gas, you have so much vapor in that environment that's going to be hard for the plant to exchange because you already have a lot of water in form of gas. So more moisture, less transpiration. And on the other hand, if you have a dry environment, a very dry, you're going to lose more water to this dry air outside. All right, this is how it works. Okay, so here we finished the standard level component. If you're a standard level student, you can stop by now, okay? If you're a higher level student in biology, you continue because we're going to cover the additional higher level topics for this gas exchange. Okay, so the first idea is adaptations for hemoglobin, okay, in higher level. It's not that complicated. So hemoglobins are typical cells. They're red blood cells. They're involved in in the transport of gas and gas exchange in the body. And they don't have a nucleus, and they have this concave structure to increase the surface area. And they can transport a lot of oxygen, the hemoglobins that are present in its surface, okay? So the hemoglobin is a protein that has a quaternary structure. So it's made of 4 polypeptides, and they end up combining these tertiary structures. They combine to each other, okay? And they're also conjugated proteins because they have a heme group. They have the iron factor. Okay, this is what makes the blood red and with that typical smell and structure. Okay, so we have these globins combining with these heme groups. In these heme groups and in the globin, we attach the oxygen to this cell. And then this cell is going to move through your body, and it's moving through your brain exactly right now. And this is what makes you process this audio and this video that you are watching, giving you energy and oxygen to make energy. Beautiful, beautiful protein. So here we have the quaternary structure, the 2 beta tertiary structures, and 2 alpha tertiary structures with the heme groups that contain iron. All right. So here we're going to attach the oxygen molecules, and this is going to be delivered through the body. Okay. Before we finish, we have what we call saturation of oxygen. Okay, we have saturation of oxygen in the hemoglobin, right? So these hemoglobins, they are attached with oxygen, and as they move through the body, they are going to deliver this oxygen, okay? And they're gonna be consumed. And on different places, we have different concentrations of oxygen, especially as you change the height. And we have different typical saturations of hemoglobins, okay? This is why in the sea level, okay, it is very easy to have your hemoglobin saturated with oxygen, all right? But as you go up, for example, on Mont Blanc or Everest, where we have less oxygen, the saturation is less likely. Okay, so we have a linear relationship between these variables. Okay. So for example, here we have the graph of adult hemoglobin, all right? So as you go to the sea level, you have more oxygen, you saturate. If you are in higher altitudes, um, higher places, the saturation is going to be less likely because less oxygen is available to attach to the heme groups. All right. What happens? This is especially interesting when we talk about the fetal hemoglobin, because the fetus inside the womb of the mother, inside the uterus, is getting the oxygen from the mother. All right, so this oxygen arrives in the lung of the mother, and then it's going to the blood and finally arriving in the fetus. So the fetal hemoglobin has more affinity to oxygen, has a higher affinity. So the saturation is even more likely than the adult one. Okay, it's just this. Okay, saturation depends on the concentration of oxygen, it depends on several factors, and the fetal hemoglobin has more affinity than adult one. Okay. Um, That's it for today. I'm going to leave this investigation and this link in which you can compare gas exchange in a simulation. Okay, you can compare vital capacity and forced vital capacity. In other words, amount of liters that a lung can uptake and expel. This is interesting to practice, what we studied so far. But I hope that was helpful. This is the higher level, this additional higher level. And thank you for watching.